Genetics and health. Definition. Genetics is the study of gene, heredity, and genetic variation in living organisms. It is generally considered a field of biology, but it intersects frequently with many of the life sciences and is strongly linked with the study of information systems. History. The father of genetics is Gregor Mendel, a late 19th century scientist. Mendel studied trait inheritance, patterns in the way traits were handed down from parents to offspring. He observed that organisms, plants, inherit traits by way of discrete units of inheritance. This term, still used today, is a somewhat ambiguous definition of what it referred to as a gene. Trait inheritance and molecular inheritance mechanisms of genes are still a primary principle of genetics in the 21st century, but modern genetics has expanded beyond inheritance to studying the function and behavior of genes. Gene structure and function, variation, and distribution are studied within the context of the cell, the organism, e.g., dominance, and within the context of a population. Genetics has given rise to a number of subfields, including epigenetics and population genetics. Organisms studied within the broad field span the domain of life, including bacteria, plants, animals, and humans. Genetic processes work in combination with an organism's environment and experiences to influence development and behavior, often referred to as nature versus nurture. The intra- or extracellular environment of a cell or organism may switch gene transcription on or off. Burden of genetic disorders. Each year, more than 3 million children born with a serious genetic defect die. Most of these deaths, 90%, occur in developing countries. In the Western world, there is 1% chance of having an inherited disease at birth. Approximately 5% of the world's population carries trait genes for hemoglobin disorders, mainly sickle cell disease and thalassemia. Over 3 lakh babies with severe hemoglobin disorders are born each year. The estimated incidence of Down syndrome is 1 in 1,000 live births worldwide. Each year, approximately 3,000 to 5,000 children are born with this chromosome disorder, and we believe that there are about 250,000 families in the United States of America who are affected by Down syndrome. The incidence of cystic fibrosis varies across the globe. Although it is severely underdiagnosed in Asia, existing evidence indicates that the prevalence of CF is rare. In the United States of America, the incidence of CF is reported to be 1 in every 3,500 births. Burden of genetic disorders in India. Burden of non-communicable disease in India. Reasons of high prevalence in India. Consanguineous marriages. High birth rate. Poor governmental support facilities. Lack of expertise in genetic counseling. Lack of improved diagnostic facilities. Classification. Classification of diseases based on their genetic basis as monogenic, Mendelian, disorders. Chromosomal aberrations. Polygenic disorders. Mendelian disorders. We observed 6 general patterns of inheritance. Autosomal recessive. Autosomal dominant. X-linked recessive. X-linked dominant. Co-dominant. Mitochondrial. Autosomal recessive. The disease appears in male and female children of unaffected parents. E.g., cystic fibrosis, phenylketonuria. Autosomal dominant. Affected males and females appear in each generation of the pedigree. Affected mothers and fathers transmit the phenotype to both sons and daughters. E.g., neurofibromatosis, adult polycystic kidney disease. X-linked recessive. Many more males than females show the disorder. All the daughters of an affected male are carriers. None of the sons of an affected male show the disorder or are carriers. E.g., hemophilia A and B, color blindness. X-linked dominant. Affected males pass the disorder to all daughters but to none of their sons. Affected heterozygous females married to unaffected males pass the condition to half their sons and daughters. E.g., vitamin D-resistant rickets, familial hypophosphatemia. Codominant inheritance. Two different versions, alleles, of a gene can be expressed, and each version makes a slightly different protein. Both alleles influence the genetic trait or determine the characteristics of the genetic condition. E.g., ABO locus. Mitochondrial inheritance. This type of inheritance applies to genes in mitochondrial DNA. Mitochondrial disorders can appear in every generation of a family and can affect both males and females, but fathers do not pass mitochondrial traits to their children. E.g., Leber's hereditary optic neuropathy, LHON. Chromosomal aberrations. Alternations in the number or structure of chromosomes. Autosomes or sex chromosomes. Numerical abnormalities. Polyploidy. Trisomy, Klinefelter's syndrome. Monosomy, Turner's syndrome. Structural abnormalities, breakage followed by loss or rearrangement deletion, translocation. E.g., in CML, or due to exposure to radiation. Multifactorial inheritance, polygenic. Influence of multiple genes and environmental factors. These include mainly the non-communicable diseases. Diabetes mellitus. Hypertension. Cardiovascular diseases. Cancers. Most diseases have a genetic component. In many cases, a single defective gene is not sufficient to cause a genetic disorder. Yet many of the common diseases of adult life, such as diabetes mellitus, hypertension, schizophrenia, and most common congenital malformations, such as cleft lip, cleft palate, neural tube defects, have a strong genetic component to their occurrence. It is thought that numerous genes each act in a small but significant manner to predispose an individual to the genetic disease. Polygenic genetic diseases are those which are caused by the impact of many genes, each having only a small individual impact on the terminal condition. Multifactorial traits are those which result from an interaction between multiple genes and often multiple environmental factors. For example, some vegetarians with acceptable cholesterol levels suffer myocardial infarction in the 30s. Other individuals seem to live forever despite personal stress, smoking, obesity, and a sedentary lifestyle. Cystic fibrosis, trisomy 21, Fragile X, hemophilia, DM, HT, heart disease, obesity, schizophrenia, asthma, RSA, TB, struck by lightning, meningococcal infection. Genetics versus genomics. Genetics. Conditions caused by an extra or missing chromosome or part of a chromosome, caused by a mutation in a single gene in chromosome or in a mitochondrion, are important to the individuals and families who have them. Genomics refers to those conditions plus discoveries from the Human Genome Project, HGP, which show that most adult-onset and chronic diseases can be partially caused or prevented by genetic factors. Environmental factors also play a significant role. Human Genome Project. In 1990, this project was initiated as a joint effort of U.S. Department of Energy and the National Institutes of Health. In April 2003, Human Genomic Project sequencing is completed and the project is declared finished 2 years ahead of schedule. Improve diagnosis of disease. Detect genetic predispositions to disease. Screening. Advice. Risk factor modification. Create drugs based on molecular information. Design custom drugs, pharmacogenomics, based on individual genetic profiles. Use gene therapy for treatment. Identify potential suspects whose DNA may match evidence left at crime scenes. Exonerate persons wrongly accused of crimes. Pharmacogenomics. The development of drugs tailored to specific subpopulations based on genes. Pharmacogenomics has the potential to decrease side effects of drugs. Increase drug effectiveness. Make drug development faster and less costly. Use of medications otherwise rejected because of side effects. New medications for specific genotypic disease subtypes. Gene therapy. Gene therapy is introduction of normal genes into cells that contain defective genes to reconstitute a missing protein product. Gene therapy is used to correct a deficient phenotype so that sufficient amounts of a normal gene product are synthesized to improve a genetic disorder. Modification of cells by transferring desired gene sequences into the genome. Delivery systems available. In vivo, delivery of genes takes place in the body. Ex vivo, delivery takes place out of the body, and then cells are placed back into the body. In vivo techniques usually utilize viral vectors, virus, carrier of desired gene, e.g., adenovirus, retroviruses, herpes simplex virus. Virus is usually crippled to disable its ability to cause disease. Viral methods have proved to be the most efficient to date. Many viral vectors can stably integrate the desired gene into the target cell's genome. Ex vivo manipulation techniques as electroporation, liposomes, calcium phosphate, gold bullets, fired within helium pressurized gun, retrotransposon, jumping gene, human artificial chromosomes. Preventive and social measures in genetics. Health promotional measures. Eugenics, positive and negative eugenics. Euthenics. Genetic counseling. Nutritional genomics. Specific protection. Early diagnosis and treatment. Rehabilitation. 1. Health promotion measures. Eugenics. In 1883, Francis Galton coins the word eugenics from the Greek for good, eu, and born, genics. We define it as the science of improvement of the human race through better breeding. It is of 2 types, positive eugenics and negative eugenics. Positive eugenics promotes marriage and breeding between people considered desirable, and though a positive eugenist may view certain persons as undesirable, they will not initiate in such practices as non-voluntary sterilization, genocide, active euthanasia, or any other forms of violence. In fact, as these eugenists say that, the defective will always be with us, since people with hereditary defects come from the general population and not strictly from other defectives, there is no logical way to get rid of them. By promoting marriage and unions between desirables, it may be possible to increase the national, even universal, average in the course of 4 or so generations. Negative eugenics. Negative eugenics, improving the quality of the human race by eliminating or excluding biologically inferior people from the population. This goal required severe restrictions on reproductive rights, for those with defects had to be kept from reproducing, if necessary, through the forceful sterilization. Elderly and sick people killed under Hitler's policy of eugenics. Euthenics. Euthenics is a science concerned with improving the well-being of mankind through the improvement of the environment. Mere improvement of genotype is of no use unless the improved genotype is given access to a suitable environment, which will enable the gene to express themselves readily. E.g., children with mild mental retardation when placed in an encouraging environment showed improvement in their IQ. Euthenics measures must be comprehensive to include physical, intellectual, social and cultural components whereby genetically disadvantaged individuals can achieve a reasonable degree of development. Measures must be aimed at improving the environment in order to improve health, appearance, behavior, or the well-being of society. Genetic counseling. The genetic is done by a genetic counselor who is a health professional who is academically and clinically prepared to provide genetic services to individuals and families seeking information about the occurrence, of a risk of occurrence, of a genetic condition or birth defect. The counselor provides client-centered, supportive counseling regarding the issues, concerns, and experiences meaningful to the client's circumstances. The genetic counselor communicates genetic, medical and technical information in a comprehensive, understandable manner with knowledge of psychosocial and cultural background of each client and their family. Prenatal genetic counseling. Preconception counseling, if learned prior to conception that female and or her partner are at high risk of having a child with a severe or fatal defect. Options will be pre-implantation diagnosis, when eggs that have been fertilized in vitro, in a laboratory, outside of the womb, are tested for defects at the 8-cell, blastocyst, stage, and only non-affected blastocysts are implanted in the uterus to establish a pregnancy. Using donor sperm or donor eggs. Adoption. Indications for prenatal diagnosis. Advanced maternal age. Previous child with a chromosome abnormality. Family history of a chromosome abnormality. Family history of single gene disorder. Family history of neural tube defect, NTD. Family history of other congenital structural abnormalities. Abnormalities identified in pregnancy. Other high-risk factors, consanguinity, poor obstetric history, maternal illnesses. Prenatal screening for genetic disorders. Invasive testing. Amniocentesis. Chorionic villus sampling, CVS. Chordocentesis. Pre-implantation genetic diagnosis. Fetoscopy. Non-invasive testing. Ultrasonography. Maternal serum AFP. Isolation of fetal cells from maternal circulation. Process of genetic counseling. 1. Beneficiaries, individual or couple. Have affected child. Are carriers. Have genetic disease in family. Have recurrent abortions. High maternal or paternal age. Exposed to a mutagen or teratogenic. Are consanguineous. 2. Reaching an accurate diagnosis. Family history. Physical or clinical examination. Cytogenetic studies or radiology. Laboratory or DNA analysis. 3. Estimation of recurrence risk. Family pedigree. Applying various methods. Risk calculation. Mendel's law. 4. Genetic counseling. Available options. Risk calculations. New developments. Disease course. Treatment availability. 5. Decision making. Knowledge of disease recurrence. Available options. Family pressure. Religious beliefs. Social status. Economic status. Community influence. 6. Gene therapy. In vivo. Ex vivo. Nutritional genomics. The study of how different foods can interact with particular genes and alter the disease's process, as in type 2 diabetes, obesity, heart disease, and some cancers. It's beneficial as well as harmful up to some extent. As foodstuffs containing antioxidants. Potential benefits. Increased focus on a healthy diet and lifestyle. Motivate positive behavior change. Improved health and quality of life. Focus on prevention. Decreased morbidity and premature mortality. Reduced healthcare costs. Identify subgroups who might be particularly responsive or resistant to environmental, dietary, intervention. Better understanding of the mechanisms involved in disease susceptibility. Potential harms. Focus on specific nutrients or foods. Attention is drawn away from other modifiable risk factors. Decreased use of other services. False sense of security. Misleading claims. Increased costs associated with personalized diets and designer foods. 2. Specific protection. Protection of individuals and entire community against mutagens such as X-rays and other ionizing radiations. Patients undergoing X-ray examination should be protected against unnecessary exposure of gonads to radiations. Prevention of Rh hemolytic disease of newborn by immunization with anti-D-globulin. 3. Early diagnosis and treatment. Post-implantation, received a diagnosis of a severe or fatal defect after conception. Possible options may include preparing family for the challenges they will face when they have a baby. Fetal surgery to repair the defect before birth. Surgery can only be used to treat some defects, such as spina bifida or congenital diaphragmatic hernia. Most defects cannot be surgically repaired. Ending the pregnancy, for some families, knowing that they'll have an infant with a severe or fatal genetic condition seems too much to bear. Referral to cardiologist to discuss heart surgery, and a neonatologist to discuss the care of a post-operative newborn. Carrier screening for thalassemia and hemoglobinopathies should be offered to a woman if she and/or her partner are having a positive family history. Ideally, this screening should be done conceptionally or as early as possible in the pregnancy. 4. Rehabilitation. Early referral of children with genetic disorders which are known to cause physical or mental disability. Research in India. In India, National Centre of Applied Human Genetics was started in March 1980 at the Institute of Medical Sciences, Banaras Hindu University, followed by establishing human genetics in a university setting at Jawaharlal Nehru University since March 1989. In April 2002, Human Genetics Laboratory of JNU was announced as National Centre of Applied Human Genetics. NII, National Institute of Immunology, New Delhi. NCCS, National Centre for Cell Science, Pune. CDFD, Centre for DNA Fingerprinting and Diagnostics, Hyderabad. NBRC, National Brain Research Centre, Mansar. Institute for Bioresources and Sustainable Development, Imphal. Institute of Life Sciences, Bhubaneswar. Bharat Immunologicals and Biologicals Corporation Limited, Bulandshahr. Indian Vaccines Corporation Limited, Gurgaon. These institutions are equipped with world-class instrumentation and have been provided with highly competent human resources. Genetic Services. Medical Termination of Pregnancy 1971 lays down a legislative framework of application of genetics science. PCPNDT Act 1974. Sex-selective abortions need to be eliminated by implementation of PCPNDT Act. Antenatal clinics provide opportunity to educate individual and family on genetics. Screening of antenatal mothers for Rh grouping. Avoidance of teratogenic drugs and radiation. Promoting immunization against rubella before pregnancy is preventive genetics. Contraception for limiting the size of family and bearing children at the right age is an example of positive and preventive genetics. Improving nutritional status and consumption of iron and folic acid. Use of ultrasonography to detect defective children is essentially a good screening activity. Early Childhood Development Services in ICDS program by providing environmental stimuli is an attempt to realize full potential of genetic endowment of young children. Summary. Genetics is a study of inheritance dealing with the transmission of hereditary characters from one generation to another. Human genetics is concerned with the inheritance of human traits and their relationship to the human health. Deals with the hereditary disorders and provide key to their prevention and control. Conclusion. Genetics is the branch of medicine which has huge potential for researches, which will help in prevention, early diagnosis and cure of diseases. As most of the diseases have a more or less of genetic component, it will help in creating more productive and disease-free society. Please like, subscribe and click the bell icon. Thank you for watching video.